to our core facilities seminar series. Today we'll be featuring the acquisition, processing, dissemination, and molecular analyses of tissue for your research studies available through the Histology core at PVLSI. For those of you who do not know me, I am Lisa Korpieski, the Director of Communications and Marketing here at the Institute for Applied Life Sciences. In today's seminar, you will be hearing from Dr. Sallie Schneider, the Director of the Biospecimen Resource and Molecular Analysis Facility, BRaMA, in the Pioneer Valley Life Sciences Institute at Bay State Medical Center. We are hoping that with these bi-weekly seminars, you will discover what great resources that the centralized UMass Amherst core facilities offer to our campus community, the New England region, and beyond. And next, just a few housekeeping items. This seminar is being recorded. If you miss any part of this seminar or would like to forward it to someone who cannot attend, there will be a replay of this and all previous seminars in this series on our website. I will put a link in the chat. I recommend you set your view mode to speaker. Please stay muted during the talks. We will save the Q&A until the end of the presentation. During that time, I welcome you to unmute yourself and ask your question. You can also put your questions into the chat, which I will be monitoring. Thank you. Next up, I would like to introduce you to Andrew Bernard, Director of our UMass Amherst core facilities. Andrew?
Thank you, Lisa, and thank you all for joining today. My name is Andrew Bernard, and I'm the Director of the centralized core facilities here at UMass Amherst, as Lisa mentioned. We're excited at how well the seminar series has been received, and we look forward to sharing more information on the great resources we have available. Today's facility is not one under management by the Institute for Applied Life Sciences, but one we work closely with. While my primary mission is to advocate for the 30 centralized cores, I appreciate that there are a significant amount of facilities throughout campus and in the area that the research community can take advantage of, and I hope to share more of them as we move forward. This is now our 7th seminar of the semester, with several more to come, and we are busy planning for the spring semester. If there are cores or specific technologies you would like us to feature, whether they be part of the centralized portfolio or not, please let us know by filling out the form that will be circulated after the semester. For those who haven't interacted with us much, the UMass core facilities are open to anyone from undergrads through senior scientists, regardless of affiliation, including researchers from other academic institutions and commercial partners. From sending samples for analysis to designs for 3D printing to becoming a trained user on advanced imaging instrumentation, there are ample opportunities for engagement, no matter your level of expertise. Additionally, there are many opportunities to fund your work in the cores. For UMass users, there are several seed funding programs and core credits available to all IELTS faculty members. For our external users, the UMass Innovation Voucher Program subsidizes usage for small companies based in Massachusetts up to 75%. Over the last 2 years, across the 5 UMass campuses, we have awarded more than 450 vouchers worth more than $5 million. If you happen to be a Mass Bio member, there is also a 10% discount you may be eligible for. We're here to be your partner and help expand your research productivity. Feel free to reach out to me directly if you have any questions, or to any of the core facility directors individually if you'd like to gain more information about their capabilities. It's now my pleasure to turn the reins over to Dr. Sallie Schneider. As Lisa mentioned, she's the Director of the Biospecimen Resource and Molecular Analysis Facility at PVLSI. I'll ask Dr. Schneider to give a brief introduction of herself, and then we'll get going. Thank you.
All right. Can everyone hear me?
Yes.
Okay, good. All right, so I think I was, for whatever reason, the slides are not progressing. Huh. All right, here we go. I'm sorry about your slides there, Andrew. All right, so my name is Sallie Schneider, and I am the Director of the Biospecimen Resource Facility. It's probably one of the best kept secret core at UMass because I'm down in Springfield. I actually got my PhD at UMass under Barbara Osborne, did my postdoc out at Harvard Medical School with John Blennis, and then returned to the area for family reasons. And I've been working down at the Pioneer Valley Life Sciences Institute since 2002. This is our team down there, obviously myself, Dr. Kelly Gregory, who also graduated from UMass. She works with me as sort of a collaborator and team member. Jennifer Delansky is the histotechnician. I do not have a picture of her, I'm sorry, Jen. Jen has her master's from the University of New Hampshire, and she would be the one you'd be directly interacting with if you would be interested in some of our histology needs. And Jerry is the scientific director. So what is the Pioneer Valley Life Sciences Institute? Really, this was a collaboration set up between the University of Massachusetts Amherst and Bay State Health back in 2002, and it was designed to try and help increase and facilitate translational research. So at that time, as a person who was working at UMass Amherst, you realized very quickly that you could do a lot of mouse stuff, you could do a lot of cell line stuff. It was very difficult to know who to talk to about possibly getting human specimens. There are certainly some clinicians who are interested in research, and there are some that are just a little too busy to do it. And so what was thought was that by sort of merging this together, and the first groups all centered on breast cancer, because there were and still are several faculty members and clinicians who are interested in breast cancer research, that we could help each other this way. And from the Bay State end of things, that they might have some wet lab space and have someone who would help train surgical residents, pathology residents, people who might want to do research that way. So I am, as I said, director of what I'm going to refer to as BRaMA from now on. And what BRaMA does is to work with the surgeons and the pathologists at Bay State to acquire tissues as needed for research. We also offer a range of services through our histology core, and I'll show you pictures of those in a minute. And then hopefully, allow everyone to have access to this tissues and the data, if they have IRB approved protocols. Our capabilities are, which will hopefully add value and help to you guys, should support preclinical research. So what do we have? What can we do? Certainly we can help with processing and paraffin embedding. We have some lovely equipment to do that. I know there are a couple labs on campus that might have these, but not everyone knows who to talk to. And I have a technician who can do it for you if you don't want to do it yourself. So there's, as I said, processing, paraffin embedding, there's tissue sectioning, either frozen or paraffin embedded. We have automatic tissue staining for either H&Es or immunohistochemical analysis. And we have a laser capture microdissection instrument. I know you have one on campus, but if that one's busy or if you need someone to help you with it, we can do that too. Several of these instruments, in particular the microtomes and the microdissection instrument, are available if you want to do it yourself. For an hourly rate, we can train you how to do it, and you can come down and take care of it yourself. So I really wanted to spend the majority of this talk, though, on the other part that probably most of you don't know. And that's on what I can do for you tissue registry-wise, for those of you who might be more interested in translational research. As I said, our main focus at the beginning was in breast cancer. And so this is the largest and the most well-established registry. It has almost 1,300 participants so far. Not all of them have had enough tissue to be able to donate it, but they have data. And so we have an honest broker who gathers all this data. We have a large questionnaire that will provide data on reproductive history, health history, some sort of, well, like whether they're drinking or smoking or take a lot of ibuprofen or things like that. So there's a lot of information there collected from these 1,300 participants. And this is added value. So if you have any interest in how demographic affect breast cancer, this registry might be able to help you. Now, from some of these women who are having reduction mammoplasties or mastectomies, or in some cases lumpectomies where the tumor was over 1 centimeter, then we were able to collect tissue. So we work with the surgeon and pathologists, and we get the tissue that is not needed for diagnosis and can be donated for research. And so to that end, we have about 435 different tissues that have been donated. Most of them have matching bloods. We have isolated primary normal breast epithelium from over 250 of these. And so these can all be used for research. And in a minute, I'll go through how these have been used. But we have two other registries, and these are newer registries that I just want to make you aware of. One is very new, and this is our metastatic blood registry. So if you were interested in biomarker research, you know, if you had something that you thought could identify someone who might respond better to one treatment than another, or something that might come up after treatment, then this might be a registry that you might be interested in the future. I mean, so far we only have 7 participants, but blood is taken from those participants before they start treatment. And then every 3 to 4 months after they've been evaluated, you know, there are notes on clinical response, and then if they've been changed to a different therapy. So it's an interesting registry that's just starting to grow. The other one we have is an ovarian tissue registry. And to date, actually it's 72 because we got one yesterday. 72 women have been registered, and we have, again, tissue and blood. This is almost invariably tumor tissue in this case, from 36 patients. So what do we do when we get the tissues in? Well, we always fix and we always freeze. Fix for paraffin embedding and freeze. But we also can do explant cultures. We digest the tissue for purification of the breast epithelial cells. And more recently, we've been making sure to freeze back organoids and spheroids where possible from the tumor and sometimes the normal tissues. For the cells that we grow in 2-dimension, we grow them out for 3 passages and we freeze back 9 vials. We also have blood from most of these women. In this case, we freeze back the plasma, the buffy coat, and we take blood spots on FTA paper. And my lab so far has been using that for STR analysis to compare back to the cell lines so that we can confirm the cell lines are from the actual individual. So now I just wanted to take a few moments to highlight some of the really cool research that has been happening at UMass using these tissues. And maybe now that you all hear about these tissues, maybe you'll think of, maybe we'll spur something that you might want to use it for. But the first thing I wanted to talk about were these explants. So this is tissue that is diced or sliced up fairly small, but that we place on gel foam sponges and media. So they're at the proper air-liquid interface. And what's neat about this is they have all the normal heterotypic interactions that are supposed to be there. So the other neat part is that because we are doing it in small amounts, we can treat them in different ways and compare within one individual how they respond to different things. So this was taken from Karen Dunphy's paper, and she was obviously looking at the effect of estrogen and radiation on these responses. And you can see in her picture here, you know, she's likely to be able to put out 4 slices per plate, and that allows, you know, 1 for fixation and 3 for maybe RNA. But it gives some very powerful results. So she just recently published this paper, and as I said, her focus was on estrogen responses because estrogen has been known to increase the risk for developing breast cancer. And what she found were looking at RNA levels that in these reduction mammoplasties, that estrogen significantly increased several genes that were expected to increase. And actually, interestingly, we didn't see it significantly change in the paras. And maybe that agrees with what we know because generally nulliparous women are at higher risk for developing breast cancer than paras women. She was also able to use the formalin-fixed paraffin embedded sections to look for R loops. And so R loops are induced by estrogen in all these women. But again, there's variation. And so the question is, does this variation maybe suggest a difference in susceptibility to breast cancer? Another person who has used the fresh tissue for these reduction mammoplasties is Genwalu in, I think he's nutrition. And so he had been studying obesity and knew that obesity tended to increase Wnt signaling. And so he was interested in determining whether Tnf alpha, in particular, one of the inflammatory cytokines that would be upregulated in obesity, might be controlling the Wnt signaling. And so what we set up for him were these explant cultures with either a recombinant TNF alpha or a neutralizing antibody to TNF alpha, and we're able to demonstrate the downstream Wnt signaling was affected. And then finally, with the explants, Dr. Kelly Gregory has been working with us on a UO1 to understand endocrine disrupting chemicals on their breast cancer risk. And our interest really has been in the impact of the immune system, epithelial, the crosstalk that exists. And so for this project, one thing she had to do was to show that she could polarize macrophages within these explant tissues by exposing them to LPS and interferon gamma towards M1 macrophages, or IL-4 and IL-13 to polarize it towards M2-like macrophages. And I'm just showing a little bit of data with these particular markers that would mark either, like the HLA-DR alpha would be M1 and the CD209 in this case would be M2. But you see the variation, and that variation is very interesting to us because not all women who get exposed to the same thing develop breast cancer. So what is it? Is it a timing of exposure? Is it something within them genetically or a microenvironmental thing that controls it? It's all very interesting. And the rest of the paper, she was able to show that some of these endocrine disrupting chemicals would appear to polarize these macrophages towards M2, which might increase breast cancer susceptibility. All right, so formalin-fixed paraffin-embedded tissue, we have a lot of. And while it is static, you can answer a lot of interesting questions. And so I wanted to highlight two of these studies. The first one is by Kathleen Arcaro, who is interested in methylation and how these early changes, epigenetic changes, might increase breast cancer risk. While she is primarily focused on breast milk, this was an interesting foray into breast cancer. And so what we were able to do for her, because we get a lot of bilateral mastectomies, is able to have her laser capture microdissect out tumor compared to an area within the same breast that's at least 3 centimeters away in normal tissue, or normal tissue from the contralateral breast. And why is this important? Well, there was a question that there might be a field cancerization near the tumor that might lead to increased risk. There might be changes that might increase your risk for redevelopment of breast cancer. However, some of the data suggests that maybe people were picking up tumor cells that had migrated away. So there was some question of it. And so she was able to use her methylation analysis to compare these two normal tissues, the ipsilateral and the contralateral, to the tumor. And she found some interesting results. I mean, the ipsilateral and the contralateral normal were different. There were different number of DNA is methylated, and that the tumor, you saw this progression, particularly at the shore regions, with more methylation happening there. Sorry. So it's a very exciting way to be able to use some of our tissues that we have here. Joe Jerry has also used these formalin-fixed paraffin-embedded tissues. This is, in this case, to look at very early lesions, pre-malignant lesions, these atypical ductal hyperplasias or lobular neoplasias. They're very small, but many women will be diagnosed with them. And then the question is what to do. I mean, you usually just watch them. The women will be at increased risk. If you could identify the important changes that occur in these lesions, you might be able to find ways to prevent breast cancer or identify the women who are more likely to go on. So this was, I think, founded by the Avon Foundation. It was Joe and Grace McCurry Judson. And so what they did was look at they were able to laser capture microdissect out these early lesions and compare it within the same woman to normal epithelial from a different area of the breast. And with both these studies, we had a lot of help from the pathologists at Bay State. So it's very exciting to be able to have these particular collaborations. And what Joe was able to find after he looked at gene expression analysis was a 99 gene signature that really predicted the preneoplastic lesions. It was enriched for estrogen receptor, as we would expect. And interestingly, it also showed a consistent downregulation of a Wnt antagonist that my lab's particularly interested in, called secreted frizzled-related protein. We have used the frozen tissue for a number of studies. You might remember Dr. Melissa Troster. Now she's down at UNC. But a lot of the studies that Melissa and Natalie Shaw and Peter Shields do require these big groups because the variations in humans are huge. So you really need to look at a lot to understand how maybe demographics might relate to genes that are changed and that it could affect breast cancer risk. So while I'm not going to go into each of these studies, I think they have found it to be very valuable to have access to some of these frozen tissues. The one I do want to highlight is this one, and this is by Catherine Reeves in the School of Public Health. And she was interested in bisphenol A exposures. Now, it had been shown that bisphenol A was expressed in a lot of breast milk in maybe 80% of women or something like that. So that seems like a lot of exposure, but she was interested in whether within the fat of the breast that it, what were the levels there and how did it correlate with whether the women had cancer or not. Now, the reason I'm highlighting this was because this really took a concerted effort with me and the pathologists and the clinicians, because what you don't realize is how much plastic are in those operating rooms. So you have the chance for exposure to BPA, even as the tissue is being taken out. And so we need to minimize it all. And it was really an excellent collaborative effort between all of them to be able to collect these clean specimens, get them back, bring them in glass, and so that she could analyze it. And while it was a small study, it suggested that maybe in the women who had cancer with high BMI, there were higher levels of BPA. So I told you that we've been isolating normal epithelial cells, and I just wanted to show some of this data here. This is by my graduate student, Stephanie Morin, who has been analyzing these cells as part of one of her studies. And what we find is that in the early passage, they aren't pure epithelial cells, but by passage 1 or passage 2 with differential trypsinization, we get fairly pure epithelial cells, but they are largely basal in phenotype. So they express cytokeratin 5 and cytokeratin 14. But that's okay, they're still completely normal cells, and they will grow out to about passage 8 without immortalization, and we have played with a few manner of immortalizing them, but I like the primary cells the best. And in this case, we chose a panel of them and compared them back to some of the cells that you might see in the literature currently, like MCF10As or 76N terts, for their ability to, to respond to certain pathways. So in B here, for example, we are looking at aryl hydrocarbon receptor activity using a reporter construct, and the BP3, the benzophenone 3 that we're looking at really didn't activate it. However, PCB, as would be expected, did. And there is a degree of variation between these women, and actually it's way higher in my primary lines than it is in the established lines. And so that might make this particular panel very interesting for those people who are interested in aryl hydrocarbon receptor activity. We also looked at whether they were responsive to TGF-beta, like in C, or Wnt3 in D, using reporter constructs, and we also see variation there. So it is important to understand that women vary a wide variety, and that these variations might contribute to breast cancer risk. And so we feel like this panel could be valuable to some. We have only used them for a publication once, and that's this publication here with Nagendra Yadava. And he was really interested in the cellular bioenergetics of our cells, in particular if it changed between women who had cancer and women who did not. And so what we did in this case, we took the cells from the non-affected breast, so the breast that didn't have the breast cancer, because we didn't want that to change anything, and compared it to normal cells from women who had undergone a reduction mammoplasty. And what we saw was that there was this pyruvate-induced response that seemed to be different. Now, we don't know for sure what is happening here, but it is a really interesting observation and something that maybe could contribute to differences in susceptibility. But it was a nice use of the cells, and so I wanted to share that with you. Sue Sturgeon has used was one of the only ones who's used the blood from this particular registry. And what she used it for was to look at whether there was a difference in methylation between the blood and the breast. So many people who do a lot of exposure research use blood, changes in methylation in the blood as their measure, because you can get to that. I mean, it's not that invasive, whereas obviously taking a biopsy from the breasts would be. And since we had both of these, she was able to compare the methylation levels between line-1 and SAT2 and ALU and RAS-SF1. And what she found was actually there was a very poor correlation between the two. And so what that did, while it was a small study, it should give one pause, that you really have to check these things before going on with them. All right, the last part I want to talk to you about are spheroids and organoids. This is sort of a new addition to what I've been collecting. Why you might want to know about them, you might have gotten dinged on it in grants. I don't know, I certainly have. But what they have found, if you are looking at, say, new therapeutic agents, when they look at these spheroids or organoids that are cultured in 3D and compare them to cells cultured in 2D, that the 3D responses to the therapeutic agents mimic what happens in vivo much, much better. And so because of that, we have been working on trying to develop the system. This particular figure actually came from the Brugge lab. They were looking at normal mammary epithelial cells. They were able to show nicely that they form these lovely sort of round structures that have basal cells on the outside and luminal cells. They have the luminal progenitor as well as the mature cells. They express the estrogen receptor. So it's very nice. And that the women who are more cancer prone seem to have shifts in the numbers of the progenitor cells there. We have been really trying to work hard to get this going in lab. I talk about spheroids in this case. I'm going to refer to spheroids as slightly larger clumps of cells. These are cells that we've digested with hyaluronidase and collagenase and then collected. Slightly larger, still under 100 microns, but only grown in culture for a week, because in that time, we can still see other cells are there. And this might be very interesting to the people who are interested in the effects of immune cells, or the crosstalk between the two as we are. I can show here that there are T cells, there are macrophages. If you look at the CD68, CD45 will mark hematopoietic cells in general. So using all these methods, we feel good that there are immune cells that are carried along. When we grow normal cells, we get these lovely little lobular-like growths. And actually, I had been tasked with growing these way back at the beginning, not for this reason, but because people wanted to look at gene expression in cells that hadn't touched plastic, but they never used them. So I had them, and they were frozen for years, and we brought them back. And I think 8 out of 10 of them actually came back to form these lovely structures. So I have quite a few. If anyone was interested in growing normal mammary organoids, I have them frozen. but the tumor organoids, and now I'm going to talk about organoids as cells that can be propagated and grown, and they're from tumors, obviously. These might be more interesting in therapeutic research. And I've picked these two papers because we're trying to characterize these cells that we have growing. This one on the left-hand side actually came from Amy Black in Joe Jerry's lab, and I just like, it's beautiful. So she was looking at cytokeratins. On the right, we're looking at ER alpha. This is from our lab. It was from an ER positive tumor, so we were really happy to see that it retained ER positivity. But it would allow us to look at different therapeutics depending on what receptors are being expressed. We've also stained for like active caspases because we were interested if certain therapeutics were inducing apoptosis and proliferation. We can use Ki-67. So this is an example. These organoids are, this is a particular organoid that propagates like crazy. I've been growing it for months and it just expands and expands. It's lovely. So these were the organoids. They're actually mostly hollow and they exhibit some bridging. And there wasn't a lot of proliferation in these control organoids. You can see here in the Ki-67 label here. But we exposed them to conditioned media from macrophages, and now we start to see more proliferation over here. And if we include our endocrine disrupting chemical, now the organoids appeared to get larger, they started to fill in, we see more proliferation. So this is all preliminary data. I really just use it to show you, to highlight maybe how exciting these organoids could be, and maybe they might be of interest to you to have and grow, and I can supply them. We isolated RNA from these. In this case, since we're interested in the crosstalk between the macrophage and the epithelium cells, we were looking at CCL2. We can see that exposure to the conditioned media increased CCL2 expression. And then we were able to go back, since we had, Kelly for her paper had done a whole bunch of explant cultures exposed to BP3. We were able to go back to those same explant cultures and show that there's a subset of women where CCL2 is increased. And while it doesn't do it across the board, maybe it's these type of women who are more at risk for developing breast cancer. So in summary, I hope that by giving you these highlights, you can see how there is a great potential for biomarker research or personalized medicine, genome-wide analysis that could happen with any of these registries. And hopefully I've highlighted some of our processing. I meant to mention before, while it looks like we do mostly human and mouse, Jen has processed embedded sections stained, alpaca, frogs, ducks, skate, dogfish, bottlenose dolphin. So we do a whole bunch of different things. We would be delighted to help you with anything that we can. So yes, we can help with the unique access to some of these things. We have the staff, if you don't have the staff, to do some of this work for you. I am delighted to write letters for your grants. If that helps you at all, just reach out to me. I'm hoping we're working with Joe and Andrew to try and identify a spot on campus where things can be left, maybe a deli case, shelf, or freezer. So, because I recognize that the biggest problem is that I'm down in Springfield and you're up in Amherst, and that you guys don't have the time to drive down, but I could guarantee I could drive up every Friday and pick up whatever you need and return it to you by the following Friday. And if you need it more expedited, we could talk about maybe courier services, or you could drive down. So that's it. Thank you very much. And I am delighted to take any questions people have.
I welcome anyone to unmute themselves and just ask your question directly to Sallie.
It was either that clear or that boring. I'm not sure which. Let's see.
Sallie, maybe I'll ask one quick one. This is Joe. And do you often work up antibodies, immunohistochemical stains for people? And do you have some that you're able to share with people and support in that way for quick turnaround on manuscripts, et cetera?
We were just working on that. We were making a list of antibodies that we have used and worked out, with the caveat that what we have found, because we've done a bunch of staining, is that while we can say it works in BALB/C mouse, doesn't necessarily mean it's lovely in C57. So we're likely to have to do a little working out, but at least we'll have a starting concentration to go from. And then we are delighted to work out any other antibodies, just a matter of cost in that case. But so yes, hopefully in the website, we will be able to put up the antibodies that we have access to and that we have worked out in our case. And then we can go from there with you guys if there are new, different antibodies you want to try.
Hi, Sallie. Nice presentation.
Thanks.
What are the mechanics of working with your group to get access to human material if you want to do some pilot studies, anonymized material?
So reach out. What I usually do is pass something through the Bay State IRB for a non-human subject research determination, which is very quick. So if it doesn't require PHI, we can get you the tissue fairly quickly. If it does, then I can work with anyone to put through some IRB protocol at Bay State.
Great, thank you.
Yeah. All the data is with the honest broker, so they control that end of things.
That was great, Sallie. Thank you. It was very, very thorough and a good overview of a lot of the projects that have worked. We do get quite a few external parties coming to us to see if they can work with our facilities. Any barriers to external companies working with you?
Not at all.
Excellent.
Bring it.
Any other questions?
Hello, I have a question. It's Francesca. Thank you for the presentation. My question is, how does the training works in this period of COVID? There's like differences or?
Yeah, we haven't crossed that bridge yet. I will have to work with Jen on that one. Do you have any training or no? And what particular?
Yeah, but just for histology, for example.
For section or staining?
Yeah, for section and staining.
All right. Actually, let me get back to you after we can sit down and have this discussion. I don't know where Bay State stands with having people come down at this very point in time, but in the past, non-COVID, people have just driven down. They have worked with Jen. We have extra blocks that, to get you used to doing it. And then we can just set you up. And once you're trained, it's, I think, $25 per hour to use some of the instruments we have.
Yeah. Okay, so we can maybe speak about this later. Yeah. Thank you. Thank you so much.
All right.
Okay, I guess that's it. Thank you so much, Sallie.
I appreciate it. Thank you for giving me the opportunity to talk about it. Thank you.
And I hope everybody reaches out to Sallie directly. I want to thank you all for attending today's seminar. Throughout the series, we will be featuring individual core facilities and their relevant technologies. Our next seminar is scheduled for Tuesday, November 3rd, in which we will be learning about the advanced spectroscopy analysis services available in our new core facility from Lily Hay, the director of the Raman IR XFR spectroscopy facility. This new core facility has the capabilities to analyze versatile organic and inorganic samples, including agricultural, environmental, food, and biomedical materials, as well as polymers and heavy metals. So save the date and we hope to see you there. Goodbye, everyone.